Hi, everyone. Happy Thursday. Welcome to our webinar for today. My name is Lourdes, and I am happy to have Jennifer Wild here with us today. She is a nurse navigator in hepatobiliary medical oncology and a clinical nursing informaticist at the University of California, San Francisco. And we're going to be talking about biomarkers today. Please, if you have any questions or comments, please enter them into the chat or the Q&A. And if you do have questions, please make sure to make them general in nature, as Jennifer cannot answer questions that are specific to your case. Now I'm going to hand it over to Jennifer.
Thanks for having me. It's great to see so many people joining the live stream. I will start sharing my screen now. And we'll start from the first slide. So again, like Lourdes said, I'm a nurse navigator at UCSF in San Francisco, California. And I'm very lucky to work with a patient population that is 100% hepatobiliary. So myself and my three physicians, we only see people who have been affected by cholangiocarcinoma or liver cancer. And so we will start this slide deck with basics. This is a little bit scientific. If science was never your strong suit, feel free to just kind of nod and take in what's interesting to you for those first 10 minutes. But you don't need to understand that part. It's more just for those who are interested in the scientific background of biomarkers. In the second section, we'll talk about the actual testing. What kinds of testing do we do for biomarkers? And how much does it cost? How long does it take to get results? You know, the really meaningful factors about the test and how it might affect you. And then third, probably the part that is drawing the most interest, we will review the current on-market drugs for some of the most common mutations. And we'll also have a slide where I've listed currently open and enrolling clinical trials for some of those markers too. And again, to remind you all, I'm not a medical doctor. I'm a nurse. And so I can't answer any personalized questions about your specific case. What I hope is that from this webinar, you will come up with questions that apply to yourself that then you can write down in a way to share with your oncologist. So I hope this will just be a thought-provoking and educational meeting for you. Okay, so first, the biomarker basics. The National Institute of Health describes biomarkers as any molecule found in your blood or tissue that is a sign of something else. So in a way, things like your white blood cell count can be interpreted as a biomarker. Your vital signs are biomarkers. But when we talk about biomarkers in the oncology world, we're talking about cancer-related molecules that come from the tumor cells. So again, scientific here. What causes cancer? At the bottom line, genetic changes are what cause cancer. All of our cells have DNA. When those genes are working the way they're supposed to, your cells are following the script for how to grow, where to grow, and when to die. Cells live out their life cycle and they're programmed to die when they're not healthy anymore. When the good genes that program this script are damaged or mutated, or if you inherit bad genes from your biological parents, then cancer can grow. So there are two main ways that these genetic changes can happen. The first one you see on the bottom left there is what we're talking about, I would say probably 90% of the time when we talk about cancer biomarkers. And these are acquired mutations. This means you were not born with these mutations. This means that because of environmental factors, something has changed in the way that your cells are doing their normal job. This can come from things like the sun. It's common knowledge that when you get too many really severe sunburns, you're at a higher risk of getting melanoma, for example. Smoking tobacco increases your risk of lung cancer. These are examples of environmental exposures that can cause mutations in the cells in these parts of your body. And we call these acquired mutations or somatic mutations. The other type of mutation, which is probably what most people are more familiar with, are inherited. You get these from your biological father or mother, um, and these are passed down generation through generation. And we call these sometimes germline mutations. But I'll try to mostly just call those hereditary during this slide presentation. Now, this is probably the most science-heavy slide. For those of you who are scientists on here, I hope you'll appreciate it. If you're not, feel free to just glaze over this one. But there are three main types of gene abnormalities that can cause cancer. The first are proto-oncogenes. These are genes that promote normal cell growth because we're always growing, our body's growing back tissue that gets damaged. Um, but when these proto-oncogenes are altered, then the cells grow unchecked. One example of this is the HER2 mutation. And so when there are an increased number of HER2 receptors, more HER2 receptors than there should be, then there's increased cell growth and tumors get bigger. A second example is tumor suppressor genes. This is a safety switch. So pretend the proto-oncogene problem starts and pretend a different part of the script recognizes, oh no, we don't want this to keep growing. Where's the emergency brake? An example of this is something called TP53 deletion. So when that gene is deleted from the script entirely, there's a failure for the programmed cell death to happen. And so cells that are damaged are allowed to continue to grow and multiply. A third type of abnormality happens in the DNA repair genes. So if all of the above have gone wrong and the fail-safes are not working, then the third and final fail-safe is DNA repair. And if these also become mutated, then the repair kit doesn't get opened, so to say. The repair kit was thrown out the window. And so additional mutations even can develop. An example of this is something called mismatch repair deficiency. So some more terminology. You might see these terms on a results report if you get a biomarker test report. Driver mutations are, they're in the driver's seat. They're the mutations that are really causing some of the noticeable tumor growth. Passenger mutations are less well understood right now in the scientific community. They often, you'll see the same passengers with the same drivers a lot of times, but right now they don't seem to do anything at all. You can kind of see passengers as just being an indicator that there might be a driver, a certain particular driver involved. Variant allele frequency tells us out of the DNA molecules we found from the cancer, how many of them are affected with these mutations? Because it's not always 100%. It's often a very small fraction. A high fraction would be considered 5–10%. If you found that 50% of your cancer DNA molecules were affected, then we would be highly suspicious that there could be a hereditary mutation involved here. And there are all kinds of classifications of mutations. There are fusions where two genes get fused together that shouldn't be, deletions where a gene gets deleted, amplifications where a gene is given more of a footprint than it should have, and so on. So, science part is over. What can we do with the biomarker information? A lot, quite a lot. This can help us confirm diagnosis. Some of you may know that approximately 15 to 20% of people diagnosed with cholangiocarcinoma are initially misdiagnosed as having pancreatic cancer or cancer of unknown primary. And so biomarkers can help us narrow down what truly is the primary cancer we're dealing with here. We can also get information about prognosis because some biomarkers are associated with a better response to treatment or a more difficult response to treatment. And therapy, different biomarkers match up with different treatments that may be better for you than for someone who does not have that biomarker. And the official stance of the Cholangiocarcinoma Foundation supports biomarker testing early on in the cancer journey. You'll sometimes see an oncologist say, well, we don't need this right now. We could do this later if the first treatment stops working. But you don't want to be at that point where you get the bad news that your treatment stopped working and now you have to wait days or weeks for a biomarker test result. So we are very highly in support of doing testing early. So the testing, what kinds of tests and how to get it. Common names for different somatic tests include biomarker, next generation sequencing or NGS, tumor sequencing, and there are other names that have varying popularity across different organizations. But these are all different names for the same thing, really, at the end of the day. Um, there are, of course, scientists who will disagree with me on that, but they're pretty much all the same thing. If you get, for example, a Caris test, 95% of the information in your Caris report is the same as the information you would get through one of these other tests as well. And these are all just different companies, different vendors that make these tests available. And they have proprietary patented differences, but the methods and the machines they're using and the goals are all the same. So again, slight differences, but for the intents and purposes of this discussion, you can think of these as all synonyms. So first we have two slides about the somatic testing. And again, this is what represents the vast majority of treatable biomarkers. Somatic testing can be from tissue. You could get that tissue from a biopsy or from a surgery. You may have had a liver biopsy where you just went in for a procedure that took less than an hour, and they use a large needle to remove some pieces from your liver. Those little pieces are often called core biopsy, and they're about the size of a pea or a lentil. They're very small, but they're big enough to see and to hold. Whereas a cytology biopsy is more common. You'll see this in like a bile duct brushing. If you have an endoscopy to look at your bile ducts, they will use a very tiny pipe cleaner looking brush. Women who've had a pap smear, it's like the same kind of brush, really. And they're just kind of scratching off flakes of cells and then putting it into a special fluid. And so you can imagine if you have the option between having something the size of a lentil or pea versus a bunch of little flakes like dandruff, you're not going to have as many cells to test. And so sometimes when you have a bile duct biopsy sent off for biomarker testing, you'll get the unfortunate result report that's only one line and it will say quantity not sufficient for testing. And this is not unusual, unfortunately, with bile duct brushing. The best option is if you were lucky enough to have surgery, then you're getting a large portion of tumor cell, sometimes as large as a golf ball if your initial tumor was quite large. And so this gives a much bigger amount of cancer cells for testing. The second option with somatic testing is through blood. You'll sometimes hear this called liquid biopsy, and it's just a routine blood draw. It's usually 2 tubes, although I have seen it as 3 tubes. And this is looking for something called cell-free tumor DNA. So this is not looking for cancer cells in your blood. Just to repeat that, liquid biopsy is not looking for actual cancer cells in your blood. Instead, it's looking for the signature. It's like finding a hair at the crime scene. You're not finding the criminal at the crime scene. You're not finding the person there, but maybe you find a hair. And so that's what this is analogous to. We're looking for the DNA particles that are shed and dropped by the tumor cells into your bloodstream. Then these vendors use special techniques to remove those DNA samples from your blood and to analyze them and sequence them. There are still some, I would say, misconceptions in the oncology community setting that liquid biopsies aren't good enough. And I would say 10 years ago, arguably that was true. They were not, the techniques then weren't what they are now, but they are now very good and very widely used, very widely accepted and respectable techniques if you don't have a good biopsy to use. Now, germline or inherited testing, this is from blood or from a cheek swab. It sometimes is called normal genetic testing, and I think that's because people think of genetics, they think of their inherited genetics, so that's what's considered normal. Um, there are many vendors who do this as well. You can see them listed there. But reminder, cholangiocarcinoma is mostly affected by somatic acquired mutations and not so much by inherited ones. They can occur in cholangiocarcinoma and they can be targetable mutations of interest, but the vast majority of people are more impacted by acquired mutations in cholangio world. Most of the time, your oncology team is just ordering somatic testing for that reason. But based on your health history, your family history, they might order both somatic and inherited testing. How to get it done? First, you need to discuss it with your oncology team. They'll place an order either to their own biomarker lab if they have one, or to an outside vendor. Then you'll need to submit the specimen. If it's a pathology specimen from a biopsy or surgery, you don't have to do anything. The clinic will arrange to transfer that specimen to the biomarker lab. If you're doing a liquid biopsy, then you'll either have your blood drawn there at your oncologist's office, or if you live in a more rural area, there are the option to do home phlebotomy, where they will send a contracted blood draw staff person to drive to your home and do the blood draw there, which is very convenient. Most vendors offer it at no cost to you, and it usually takes about 2 to 3 days to schedule someone to go out to do the blood draw for you. Insurance and billing, we'll have a whole slide about this, but most commercial carriers and Medicare cover biomarker testing. Um, if you have issues with billing, you should talk to your oncology team, and we'll have a little more info on this too. Then you just wait for the results. Testing from a biopsy or surgery takes about 2 to 3 weeks from when the lab receives the sample. So remember, if your clinic is sending the sample, it might take 3 days for them to ship it. So, there can be some extension on that results time depending on transit of your sample. It's further complicated if pretend you had your surgery at hospital A, but now you've moved and you're getting your oncology care through clinic hospital B. Now hospital team B has to request the sample from hospital A, and so there can be additional logistics involved that extend the time for results. If you do a liquid biopsy with the blood draw, the results for that are much faster, and they typically take around a week. So the insurance stuff, the cost to you is usually $0 to $200. Most vendors offer a patient assistance program so that there's an income-based pay scale. You supply just one page of your tax return showing what your household income was, and based on that income, you'll receive a bill somewhere between $0 and $200. However, if you have Medicare and if you have not yet met your annual deductible, your annual maximum out-of-pocket cost, then unfortunately, there's nothing you can do but pay the cost. And this is just because of the terms of service that you all sign when you agree to have a Medicare plan, that you're going to pay a maximum annual out of pocket before Medicare covers everything else. And so unfortunately, there's nothing you can do about that. Some vendors do offer payment plans if you need to stretch out the cost over several months. But with Medicare, it can be a little bit sticky. If you're uninsured or underinsured, and if you do not qualify for patient assistance programs, So, for example, I've seen very, very wealthy people say, I don't need insurance. If I ever need anything, I have enough money to just pay for it. Then the typical self-pay cost for these tests is around $5,000. So how do you pick? Okay, I had a biopsy, I do have a sample, but which is going to be better for me? Should I send my biopsy, knowing it's the only biopsy material I have, and then once it's used up, it's gone? Or should I do a blood biopsy? If you have early stage disease, stage 1 to 2, then using tissue from a biopsy or surgery is much more reliable. And this makes sense if you think about how I was saying that with liquid biopsy in the blood, we're looking for the signature, the DNA signature in your blood. The more tumor burden you have in your body at the time of the blood draw, the more you're going to see the DNA signature from the tumor in your blood. So if you have early stage disease, only 1 or 2 tumors, they're small, they were surgically removed, there's the crime scene's not going to have as many hairs on it, you know? The criminal got in, got out, they're gone. You might not find the hair at the crime scene, so to say. Um, the downside using tissue is that you do need to have a big enough sample, like we talked about. If you only have bile duct brushings, it might not be enough, and the results do take longer. Blood can be used if you don't have a tissue sample available, or if your bile duct brushings are sent off and they're confirmed to not be adequate. Um, Blood can also be used for monitoring, although this is hotly debated at our annual conferences. If you ever have the chance to go to some of these cholangiocarcinoma conferences, like the one that we host, you will sometimes see oncologists debating this on stage, whether it's appropriate to use blood biopsy to monitor treatment and responses. So that's controversial. Talk about that with your oncologist if that interests you. But results are faster with this option. So now we're going to go to results. What do you do with the results? What treatments can match up? This is a graphic that was adapted from Dr. Valle's publication some years ago in Cancer Discovery. You'll know Dr. Valle as the Chief Medical Officer for Cholangiocarcinoma Foundation. And his research found that there are some mutations that are more common among certain subtypes of cholangiocarcinoma. So in intrahepatic, so the bile ducts that are within the meat of your liver, we see mostly FGFR mutations, IDH mutations, and also some HER2. In extrahepatic cholangiocarcinoma, the part of the bile duct that's outside of the liver, it's more common that we see the HER2 there. And in the gallbladder, HER2 as well, with some representation from BRAF. Only about 20 to 50%, I know that's a large range, only about 20 to 50% of all cholangiocarcinomas will have a targetable mutation. The other way of looking at that statistic is that about half of people who get biomarker testing for this type of cancer, unfortunately, will get a results report that says there were no targetable mutations found. So FGFR, one of the most common ones in intrahepatic. Usually we see FGFR2, but you may also see FGFR1, 3, or 4. On market, there is a drug called pemigatinib. This was the first generation FGFR2 inhibitor. I want to say it's been around for like 7 years, but don't quote me on that. It's been around for a little while. And the overall response rate is 37%, which means that 37% of people who took the drug saw a meaningful pause or shrinking in their tumors. And it helped extend people's lives by about a year and a half, according to Vogel's research. There was a second generation drug. It was known by the research name BGJ, and it was very promising, but it lost elbow room in the market to the third generation inhibitor, which is called futibatinib, or the brand name Lytgobi. The great thing about this one is that there's a little bit less drug resistance. You may have heard of drug resistance before. You'll sometimes see in the news stories about an antibiotic resistant infection going around the community. Um, and cancer is like that too. It can eventually make more mutations to say, oh, this person has found a way to fight us. We need to mutate around this fight that they're doing against us. And so you will see drug resistance occur over time in some people. Futibatinib has less drug resistance than the first generation drug, and it has a slightly better response rate as well. This whole drug family, one of the biggest concerns is potentially elevated phosphate levels. Um, Phosphate is in your blood. It should be there in some amount, but when it's too high, it can cause heart arrhythmias. The elevated phosphate can also cause some pretty gnarly fingernail changes and toenail changes where they become dry, brittle, rigid, and they can even fall off. There can be some dry eyes and vision changes, and also some irritation in the mouth and throat. For IDH1 and IDH2, this is another one of the most common mutations we see. Um, Notably, it has been shown in research that people who have viral hepatitis, such as hepatitis B or C, um, do not have IDH mutations as much as people who don't have those viruses. And IDH1 mutations and FGFR2 mutations are mutually exclusive, meaning if you have one, you don't have the other. They just do not co-occur. Um, there's one on-market therapy specifically approved for IDH mutations in cholangiocarcinoma, and that's ivosidenib or brand name Tibsovo. And this helped add an average of 10 months of life. There's also new research showing that PARP inhibitors, and this is a different class of drug for a different kind of mutation called PARP, that these might also work on IDH mutations. They're looking at this in many different types of solid tumor cancers. And there is a group that's specifically doing research right now, seeing how well this applies to cholangiocarcinoma. Now, that said, even though the research is ongoing as to whether PARP inhibitors work well against cholangio-related or IDH-related cholangio, you will sometimes find oncologists who are willing to prescribe and try olaparib, which is a PARP inhibitor, or brand name Lynparza. And it's also currently in a trial in combination with IV pembrolizumab for cholangiocarcinoma. The common side effects with this drug family are swelling of the abdomen, called ascites, nausea, vomiting, diarrhea, drop in your red blood cells or anemia, cough, and heart arrhythmia. HER2 mutations, again, these are more common outside of the liver, so in the extrahepatic cholangiocarcinoma and in the gallbladder groups. Um, and this is also more common in cholangio patients who are of Asian ancestry. There are many options for HER2. You may have heard of HER2 before in the setting of a different cancer, in breast cancer. There's something called HER2 receptor positive breast cancer. And so we've been able to take a lot of those drugs that were designed for breast cancer patients, test them in clinical trials for people who have HER2 positive cholangiocarcinoma, and prove that they can be useful for this group as well. A very popular HER2 drug is called Herceptin or trastuzumab. That's an IV infusion. There's also pertuzumab. There's a combination of Herceptin plus deruxtecan called Enhertu. Like I said, our breast colleagues have done a lot of research and they've already created an arsenal of drugs for this. And so there are many options. And there are other combinations. You can see here trastuzumab combined with another IV HER2 inhibitor. Trastuzumab combined with a chemo pill called tucatinib. Trastuzumab combined with Folfox, which is an IV chemotherapy. And there's also a very exciting trial called the Horizon trial, which is using zanidatamab. This is ongoing. It's open for recruitment. There is currently a 41% response rate lasting about 13 months. Um, and if I'm not mistaken, datamab is a very interesting HER2 inhibitor in that it is bispecific. So it's not just attacking the HER2 mutation at one point, it's attacking two different parts of the HER2 mutation. And so that's a pretty novel approach. There are some side effects with this group too. The GI things, diarrhea, nausea, vomiting. It can cause something called decreased ejection fraction in your heart. And that's when your heart is not pumping blood as forcefully as it normally would. And if you combine these with chemotherapy, then you also see all the different chemo-related side effects that come with chemo. The last mutation I'll talk about in this presentation is BRAF mutation. This is related to a BRAF protein. It's a type of proto-oncogene. Um, Only about 5% of cholangiocarcinomas have this mutation, and it is unfortunately correlated with a worse overall survival time. However, it does respond usually quite well to the drugs we have available. We use a combination of a BRAF inhibitor plus a MEK inhibitor. This is not unlike what we were talking about on the previous slides about combining HER2 and PARP inhibitors, for example. So it's two pills, Tafinlar and Mekinist. There's a nearly 50% response rate with a 14-month median average overall survival time. The main side effect with this is fevers, and I put an exclamation point because this is usually the most difficult side effect for people to cope with when they're on this medication. The fevers, they're constant and they can be they can really drain energy. There's also GI side effects, rash, and cough. Now, the slide that I'm sure everybody wants to take a picture of, go ahead. This is a sampling of what's open to recruitment as of today. You can also check the website clinicaltrials.gov. There are two trials open right now for FGFR2 mutations. One of them is using a drug called Tyra 200. The other trial is, I'm really not sure. Oh yeah, CGT 4859. That one's kind of newer, this trial, and that one's accepting both FGFR2 and 3 biomarkers. And these are both specifically for cholangiocarcinoma, and I believe they're both for subsequent treatment. So that means maybe you've already had IV treatment with Gemcis Durva, and it stopped working, and so these are considered second line options. For IDH, there are two options. Olaparib, which again is that PARP inhibitor, and durvalumab, which is an IV immunotherapy, and that's up in Toronto with Dr. Knox. There's IDH1 mutation trial with ivosidenib plus Gemcis Durva, and that's for first line treatment. So really, you're only going to know that you could be in that trial if you got biomarker testing immediately upon receiving your diagnosis before you started a different treatment. This is a first-line treatment trial only. Then you can see there are two different HER2 trials. The one here you see with zanidatamab, that's the one I was mentioning earlier on the earlier slide, the Horizon trial. That's ongoing and it's international. It's on, I think, 4 continents. There are many recruiting sites for that trial right now. I believe it's phase 3 now. It's in when you have different clinical trial phases, they grow and grow the size of the trial, the number of people to be in it. And so this is, I believe, in its final stages of recruitment. And then there's also a phase 3 trial that's using trastuzumab deruxtecan plus rilvegostomig. That one's hard to say. In HER2 cholangiocarcinoma. That one's not a first line. That one you can enroll in that potentially even if you've already had other treatment first. And that's also open in many different sites. So closing remarks, and then I'll open it up to questions and discussion. Biomarkers as the new tumor markers. Remember, I said this is hotly debated among oncologists. Some of you might already be having your blood analyzed for a tumor marker called cancer antigen 19-9 or CA19-9. This is the best tumor marker, serum tumor marker, routine marker that we have available for cholangiocarcinoma. However, it's not unique to cholangio. Other cancers, including pancreatic cancer, also express it. Um, and some people with cholangiocarcinoma, their tumors don't express this tumor marker at all. And so for some situations, it's not a reliable tumor marker. Um, if you have a driver mutation found on your biomarker testing, though, following it along with your regular imaging could be an attractive option for monitoring your cancer status. The benefits of this are that it's the future, it's precision medicine. You may have heard that buzzword before, precision medicine, and this is exactly what precision medicine is meant to be. It can be done quickly with that liquid biopsy blood draw option, and it can also identify new mutations over time, early on in their development, before their variant allele fraction is quite large. Um, and so finding them early and finding them easily with a blood draw is a very attractive option. However, there is an issue with accessibility and equity in that not all insurances cover it for this type of use. And again, not all cholangiocarcinoma patients have a targetable mutation to track anyway, and that number again is about 50%. So pros and cons to that. If you think you fall in this bucket, it's something to ask your oncologist about. And if you have not seen them, remember that we have some very nice videos that go over biomarker basics and testing. A lot of the material that we talked about in this webinar is brought through illustrated videos on our website. Here's all my references if you want to look up any of those studies. And that concludes the content. I think leaves us a good amount of time for questions and discussion.
Thank you. That's perfect. Thank you so much. We have a few questions that have come in already. For those who do not have mutations, is proteomics something worth pursuing?
Proteomics, I'm not familiar with that. Is that a vendor of a test?
They said that are there therapies targeting specific proteins?
Oh, I see. So there are a lot of potential findings on biomarker testing that they're not mutations in the sense that they're not fusions or deletions or rearrangements, but you'll just see alterations in a protein structure. And right now, there's not any treatments that match up with those proteins. If I had to guess, that's something that will come in 10, 20 years, but unfortunately, it's not something that we have made a lot of headway in the scientific community on right now.
Do you feel liquid biopsy is just as good as somatic testing?
This is a controversial answer, but I'm going to say for advanced stage disease, stage 3 to 4, yes.
Um, why is a clinical trial for IDH1 only for first line?
Well, it depends. Here, let's go back to that slide. I know that's probably the slide everyone's most focused on. Um, so this trial, ivosidenib plus durva gemcis, first line, that's because gemcis durva is the current standard of care, meaning that's the currently accepted first line treatment option, just those 3 infusion drugs, currently accepted by the National Comprehensive Cancer Network, FDA approved, international communities. This is what we've agreed as of 2020, pretty recent, or maybe 2021, that this is the best first line option. And so what this trial is doing is simply adding ivosidenib, the IDH1 mutation or IDH1 inhibitor pill. So you're taking the current standard first line treatment plus ivosidenib at the same time. If you are already getting gemcis durva or already had it, then re-challenging you with it is probably not going to do anything meaningful because we already know that you're either already on it or it stopped working for you potentially. What you could do if you already had Gemcis durva and you've found that you have an IDH1 mutation is just take single agent ivosidenib, and that's a very common approach um, that you try and potentially fail the first line option with the infusions, and then you go to just second line solo ivosidenib. So this single drug is still available to you. It's on market. Um, but this study is specifically looking at the combination of all four at once, and that's why it's only for first line.
When you've been NED and have a recurrence, should you get an updated biomarker test?
I think so. I see my physicians uh, do that. Um, and that would be most likely a liquid biopsy because if you have recurrence, um, you might have had a new biopsy, like a tissue biopsy. Say you had a new tumor pop up in a lymph node or bone. You might have that biopsy to confirm that it is the cholangio coming back and not a different second new cancer. So if you had a biopsy, you could send that off to biomarker testing. But if you had recurrence and all signs point to it being cholangio coming back, then liquid biopsy is probably the fastest thing to do.
Are you finding that reoccurrence has the same mutations as the original tumor? And do you find liquid biopsies beneficial for NED patients? Somebody else was asking the same question.
Well, when recurrence happens, it often has different mutations. So think about how you might have had your initial diagnosis, you might have had initial treatment. Um, that treatment may have involved targeting a certain biomarker at that time. And so maybe that biomarker has been quelled. Maybe it's been eliminated or pretty well controlled by the initial treatment, and then you go into NED status. Um, and so when the cancer comes back, it's often through some other new route. Maybe you sealed the tunnel with one of your mutations, but the cancer just dug a new tunnel and has a new mutation now. And so, yes, that is a very common finding that it's a new biomarker entirely.
If you've only done oral chemotherapy, would these trials consider that to be the first line or no?
Oral chemotherapy as first line is typically not seen in cholangiocarcinoma. It's usually used as a second line or subsequent line treatment. And again, that's because research has shown us that the best chance for your first line drug is with that combination IV gemcitabine, cisplatin, durvalumab, um, or gemcitabine, cisplatin, pembrolizumab. They're the same drug family, so you might see either one. But first line typically does not involve pills, chemo pills, but it can in trial, like for example, that IDH1 that we were just talking about.
Um, what is the difference between BRAF V600 versus a non V600?
They're pretty much all of you. I, I, I'm not a bench scientist, meaning I don't work in the lab, but in the way the BRAF protein is, the V600E, it just tells us the location on the BRAF protein where we're seeing the mutation. And the vast majority of them are V600E mutations in cholangio. Yeah, I will say in other gastrointestinal cancers, that's not always the case.
Is a molecular test the same as a biomarker test?
Most likely, yes. Again, these are all different synonyms. Go back to that slide here. There are many different synonyms for the different biomarker testing, and I've found that different organizations, different parts of the country, different parts of the world tend to favor different terminology. But yes, molecular testing can be a synonym for biomarker testing.
As it relates to HER2, I understand there are levels of expression which may or may not qualify you for a clinical trial. Can you please discuss the differences in expression of a mutation?
Yeah, I think maybe that goes back to this is a scientific question. This goes back to the variant allele frequency. So there are some studies that will only accept candidates who have a high enough VAF, meaning um, they want to make sure that at least 5% of your tumor cells are expressing the HER2 mutation. Um, or they might have a different cutoff. But again, it's considered high and targetable if it's as low as 1%. That means only 1% of your tumor cells um, are seeing that mutation potentially. And so um, there are some trials that want to only recruit those people who have a high enough representation of the mutation. And I have to think that that's because it's going to produce stronger evidence that the drug works for that mutation. Otherwise, there's too much of a factor from the cancer cells that are not affected by the mutation, and it could show that the drug doesn't work when it might actually work. You just didn't have enough of the mutation involved for it to work on.
Um, how can you have a high blood tumor marker, but a liquid biopsy still turns out inconclusive? Not enough tumor DNA found.
I think those are uh, two different terms that we're talking about. So remember, CA19-9 is the most common tumor marker that we use. Um, and that's just looking at an antigen that's on your tumor cells, a protein that's expressed on the outside of your tumor cells. Um, I guess by definition, CA19-9 is technically a biomarker, but it's not a mutation. CA19-9 is not a cancer mutation. Um, and so you could still be tracking CA19-9, and your tumor cells might still be expressing a high amount of this protein on the surface of your tumor cells. But the other mutations may not be present um, because they're just not related. These are two different characteristics of tumors. I hope that makes sense.
Do you have any information on the KRAS biomarkers or trials against KRAS mutations?
For cholangio, I don't. You could check the clinicaltrials.gov website. I know that they might have there might be some phase 1 trials for KRAS. KRAS is seen in cholangio, but it's more common in other types of solid tumor cancer. And so there are often you'll see phase 1 trials that are looking at certain mutations across many different cancers, so um I don't think any of these are first phase 1. Um, but you might find phase 1 trials that they're saying, okay, we'll accept people who have any type of solid tumor cancer. Um, then there are trials like these that are only for cholangiocarcinoma. And so you might find a bigger basket trial for KRAS.
Okay. Um, in your experience, how long does it generally take to get into a clinical trial?
It really depends. So first, it has to be open for enrollment. There are, I'm going to say, 3 statuses. A trial could be pre-enrollment, meaning that they're currently going through all the regulations and requirements before they can open. I mean, if they're pre-enrollment, it means they're going to be given the green light soon. If they're open and recruiting, you reach out and you try to get in. If it's open but not recruiting, it means the trial's still ongoing. There are still people in the trial, but they've closed to enrollment. They've reached their cap, their limit of how many patients they're allowed to have in it. And so only the people who are already in can continue. Um, if you're talking about the second category where it's open to enrollment, um, the time from saying, hey, I want to do this trial to receiving the drug can range, I'm going to say, a week to 3 weeks. And it depends on a lot of the pre-trial diagnostics. Trials always have requirements to make sure that you're fit for trial. They're going to make you do potentially a lot of blood work, a new scan. They might make you do a new biopsy if you have a tumor to biopsy. They might make you do some cardiology tests to make sure your heart is in a good enough starting point. And it takes time to do those tests. And so after you finished all those tests, after your doctor sends all that information to the study, then they give you the green light to participate. They randomize you into a treatment arm, um, and then they send the drug to your doctor's office. And so that whole process, I will say at best takes a week. I've seen it happen in a week, but more typically it's like 2 weeks or up to 3.
Okay, thank you. Um, if someone had a successful resection, the tumor was removed, it was sent off for biomarker testing. In your experience, if they have a recurrence, will that recurrence of the cancer usually have the same biomarkers, or do they usually come back different?
If that person never had any uh, targeted treatment, which they probably didn't. They may have had either no treatment or they may have had post-surgery treatment with Xeloda or capecitabine chemo pills, which is not a targeted chemotherapy. Um, so if they did not have a targeted treatment, then probably the same biomarkers are going to be expressed during the recurrence. Um, if a person had some kind of targeted treatment and then went into NED, then the recurrence might have different biomarkers expressed. Now, that said, if the recurrence uh, is a long time after the initial surgery, I personally would think it was a good idea to do a liquid biopsy if it had been more than a year or two. I would, if I was the patient, I'd ask, is it appropriate to do a liquid biopsy and see what your oncologist says?
Okay. Is the controversy using blood testing for monitoring cancer that it gives false hope as a tumor load is low?
I don't think it's about false hope because again, the let me go to that slide. Let me find it. Um, the the variant allele fraction, so again, the representation of the cancer DNA, how much of it is there. If it's there and it's above, 1 or 2%, it's worth targeting. That's pretty well agreed upon in the scientific community. The issue maybe that this person is asking about perhaps has to do with the early stage disease problem. And this is just simply a problem of the science of cancer. If you have early stage disease, you just don't have as much tumor cells to leave hairs at the crime scene in the first place. There's just no way for us to say that that test was reliable. Um, but I think that the bigger issue, the bigger part of the debate with using liquid biopsy for monitoring, um, is the use of resources that, clogging up the lab with running repeated tests for some people when other people haven't even had their first test and they're waiting every day for the result. I think that's probably a current issue that could change, but of course that costs money. All these labs have to grow then. And we do use serial biomarker testing in other cancers. It's FDA approved for colorectal cancer and for breast cancer, and possibly for other cancers too. But right now in the hepatobiliary space, it's not FDA approved for continuous serial monitoring, which is why I think maybe there's not as much funding into expanding some of those laboratories in that way.
In your experience, if someone has IDH1 mutation, but they've been on the standard of care, GemCis and immunotherapy, and they're doing well, is it unlikely that their doctor would transition them to the targeted treatment of ivosidenib?
Yeah, very unlikely. If you have something that's working, usually we want to keep people on that until it stops working. If something's working, if it ain't broke, don't fix it. That saying. You really want to save the next big thing for when you need it.
Um, just from someone. Hairs at the crime scene is a fabulous analogy.
Thank you.
Um, how does one find out what percentage of expression of the mutation they have? For example, you mentioned 1% is targetable.
Yeah, that variant allele frequency. I'm really pleased that people are interested in some of the scientific stuff. I was like, people probably won't want to know about this, but um, this is something that you will see on the full results report. So if you got your biomarker report, it's probably like, it might be only 3 pages long if there's not a lot to say. It could be 20 pages long if you had a lot going on. But somewhere in those pages, you will find VAF represented.
You mentioned that a recurring tumor may have different mutations from the initial tumor. Is that the same for the CA19-9 and that the original tumor shed, but recurring tumor doesn't shed? I think that's what they're
Well,
asking.
yeah, remember CA19-9 is not a mutation. CA19-9 is not a mutation. It is simply a protein that's on the surface of your cancer cells, and it's not unique to cholangiocarcinoma. Many different cancers have this protein, and it's just, you can think of CA19-9 as um, a normal piece of a normal tumor. Um, I'm not going to say that all tumors have it, they don't, but many, many different tumors have this as part of their normal functioning. It's not considered a mutation. CA19-9 is not a mutation. And so when we're looking at CA19-9 levels, it's not a perfect monitoring method. Um, it it may go up and down based on the tumor burden, based on how big or how many tumors a person has, but CA19-9 can go up and down regardless of what kind of treatment a person gets, regardless of what targeted mutations they do or don't have. CA19-9 is swimming in a totally different pool than the other mutations. It's a CA19-9 is a different crime scene. We'll call it that. It's a totally different crime scene. The criminal has hit two locations and CA19-9 is a totally different location.
Okay, perfect. Thank you. How does one convince their oncologist that doing another biomarker test before the current treatment stops working is worthwhile, or is it worthwhile to do that?
I think it depends um, which treatment a person is on and how long it's been from their previous biomarker testing. So if if a person had surgery and they sent that off for biomarker testing, then they were NED for a couple years, and then their cholangiocarcinoma came back and they started standard of care treatment. I would say doing another biomarker test around that time would be a good idea. Um, on the other hand, if a person just started targeted treatment, they had a biomarker test 3 months ago, it's probably too soon to do another one. I'm not going to say it's definitely too soon. I have seen I can think of them and I remember their names. There are a couple of patients over the years who had really wacky mutations, and we would check them every 3 months. And it was incredible. You would see like an FGFR2 to 3 fusion, for example, which is where you have the FGFR2 DNA molecule and part of FGFR3 gets fused onto it. And then you see that start to shrink on their tests on the variant allele fraction is going down. Then they go NED for a while. And then when it came back, it flipped. It was FGFR3, but now 2 had fused to 3. So the cancer finds weird ways to work around what we've done before. And so I'm not going to say that it's too soon, but it's probably too soon to retest.
And then one last question is, will we be able to have a copy of your presentation?
Yeah, I believe it gets posted to YouTube. And I'm also happy to put the slides on a public link to Box. If any of you are familiar with Box, that's what UCSF allows me to use to share material like this. So yeah, give Lourdes and her team a couple days to upload things, but yes, this will be online.
Perfect, thank you. And I apologize for the dogs barking in the background. But
That's
thank you so much,
okay.
Jennifer. I appreciate it.
Thank you all for having me. Thank you for your time, and I hope this has been helpful. I wish you all the best.
Thanks, everyone.